Welcome to Jeopardy for Kids. I'm your host. Let's get the show on the road. Rock with big category.
Stem cells for 100.
What makes a stem cell different than a body cell?
I don't know.
Don't worry, I got you. Body cells have a specific job. Like your brain cells think and your lung cells help you breathe. And they can only do that one job. But stem cells don't have a job yet. But they can learn to do any job. So when babies are really young and in their mommy's tummies, they have embryonic stem cells that can become any cell in the entire body. Grown-ups have stem cells too, but they can only become a certain amount of smaller related jobs. All right, pick a question now, Zoe.
Stem cells for 200.
Where do stem cells come from? What can they be used for?
There are two types of stem cells, embryonic and adult stem cells. Embryonic stem cells are cells that come from a baby when it's still in its mommy's stomach. These cells are collected really early in the pregnancy. Adult stem cells are collected from inside the bones. People feel bad when taking these embryonic stem cells because in order to collect them, babies can die. Scientists developed a way to use other types of cells instead to get rid of this problem. These cells can be used to make sure medicine for specific diseases fixes the problem caused by the disease. They can also be used to see how diseases affect people as they grow up. Finally, as I said before, these cells can be used instead of embryonic stem cells so that way no babies die from these experiments.
Good question, Rocco.
Potency for 100.
What is potency? What are the different levels of potency?
So, potency describes the ability of cells to become different cell types. The higher the potency, the more types of cells that the potent cell is able to become. The three different levels of potency are totipotent, pluripotent, and multipotent. Totipotent is the highest level of potency, meaning that it can become any cell type in the entire body. Pluripotent has the next highest level, meaning it can become any of the cells from any of the three different main groups of body cells. Multipotent has the lowest level of potency, meaning it can become a very limited range of cell types.
All right, pick a category, Allison.
Potency for 200.
How can you tell if a cell is pluripotent?
You can give a mouse a shot with cells which grow and form a big lump. You can take the lump out and see if it contains a lot of different kinds of cells. How much a cell makes is a certain thing is called gene expression. Some cells called pluripotent cells make a lot of triangles. Other cells make a lot of circles. If the cells you look at make a lot of triangles, then they are pluripotent. You can put cells into a bowl and fill it with some liquid. You don't touch it so the cells can do whatever they want. The cells grow and become more special types of cells. You can look at these cells to see if they contain many different kinds of cells.
Welcome to Jeopardy Extreme. I'm your host. Let's get started. Rocco, pick a category.
Stem cells 100.
How are embryonic stem cells different from somatic cells?
I don't know.
Don't worry, I've got you. Somatic cells are differentiated into one specific purpose, function in the body. However, embryonic stem cells have potency, or the ability to differentiate into various types of cells and functions. Differentiation is caused by various external or internal factors affecting the cell, and where these factors are located within the cell and how the cell divides influences what type of cell the stem cell will ultimately end up as. Somatic stem cells are found in the adult body, and they can have limited differentiation capabilities. However, embryonic stem cells can differentiate into all types of cells in the body. They are pluripotent. All right, Zoe, pick a category.
Stem cells 200.
What are the sources of stem cells? What are the medical applications?
Embryonic stem cells are found in the blastocyst and acquired from the inner cell mass of a fertilized egg. Ethical issues arise when obtaining embryonic stem cells because it causes embryonic death. However, adult stem cells are less useful because they have lower potency. To avoid these ethical concerns, induced pluripotent stem cells were created using methods developed by Gurdon in 1962 when he produced a frog tadpole from isolated skin cells, and further refined by Yamanaka in 2006 when he introduced Gurdon's work to the human spectrum. These cells are used to study human development as they can trace growth and observe how specific genetic diseases affect normal development. They are also used in drug development as we can test our drugs on cells that are exhibiting a disease. And when doing cell therapy for diseases, they can be used in place of embryonic stem cells.
Rocco, go ahead.
Potency 100.
What are the different levels of potency?
So, potency is a classification for the level of undifferentiation that a stem cell has. The three different levels of potency are totipotent, pluripotent, and multipotent. Through the lens of development, with the ability to differentiate in any cell type in the body, an example of a totipotent stem cell is a morula. The morula then forms the blastocyst, with pluripotent embryonic stem cells. It differentiates into three different germ layers, the endoderm, the mesoderm, and the ectoderm, which eventually becomes unipotent cells.
Allison, go ahead.
Potency for 200.
What are the methods to determine if a cell is pluripotent?
In a teratoma assay, mice are injected with embryonic stem cells which proliferate and form a tumor. The mass is taken out and analyzed to determine if it contains cells derived from all three germ layers. Global gene expression can also be examined. Potency genes are expressed at high levels in pluripotent cells, and expression goes down as commitment increases. If potency genes of the selected cells are expressed at high levels, then the cell is pluripotent. Embryonic stem cells can be put into a dish with liquid media, which allows them to spontaneously grow, commit, and differentiate without intervention. This can be analyzed to determine if it contains cells from all three germ layers.